So today we're going to do the DNA extraction lab. Okay, so you want to make sure first thing you do is put on your safety equipment. So gloves, goggles. Okay, you're going to have some equipment already set up at your table. So what you want to do first is make sure some of your glass equipment is cleaned out. So glass stir rods and also test tubes. You want to make sure you rinse them. 2 times using tap water, and then 2 times using DI water. Okay. And then what you want to do is just kind of leave them here to dry while we wait. Then you can get a strawberry. You want to put the entire strawberry into the Ziploc bag, and you want to seal it. Okay. Make sure there's not a lot of air in the bag. Once you seal the strawberry inside, and then you want to mash it with like your thumbs and your fingers, just like this, for about a minute. Okay. Once you do that, there's going to be extraction buffer at your tables. Also make sure that you guys rinse out your conical tubes, or not your conical tubes, your graduated cylinders. So again, tap water 2 times, DI water 2 times. Try to get as much water out as possible. And then you are going to measure out 15 milliliters of the DNA extraction buffer for the strawberries. Okay. Once you get 15 milliliters, then you're going to open up the bag, pour in 15 milliliters of DNA extraction buffer for the strawberry, seal it up, and then again, just kind of mash and mix around for about another 1 minute. Okay. Then you can get a cup, coffee filter, and you're going to make a sort of funnel. So what you want to do is get a rubber band, put the rubber band on the cup, just like that. Make sure you have a pocket like that, and then you are going to pour the contents of the bag into here. Make sure you don't overflow because the pocket is not that big. And then you're just going to leave this here to filter the strawberry stem and those type of things that we don't need, and let like the things like DNA drip down into the cup. Okay, so you're going to have to probably leave that there for maybe about 5 minutes or so. Okay, once that finishes draining, then what you're going to do is you're going to carefully take the coffee filter out, okay? Throw away whatever's on top of the filter. Okay. And then you are going to get a transfer pipette, okay? Disposable transfer pipette. So with the disposable transfer pipettes, there are going to be 1, 2, 3, 4 marks on the disposable transfer pipette. At the very top, there's a 1 milliliter mark. Below that, there's another mark, and in the middle, this middle mark is 0.5 milliliters, and then the lowest mark right here is about 0.25. Okay, so what we're looking for is 1 milliliter. So what you're going to do is you are going to transfer 3 milliliters of the solution in here. So what you want to do is you want to pinch the bulb and then put it inside the liquid. Slowly release until you see the liquid come up to the 1 milliliter mark. Okay. Then you are going to pull this out, and then put the entire milliliter of the strawberry DNA solution into this test tube. Okay. And you want to do that 3 times. So for a total of 3 milliliters of strawberry DNA solution. Okay. Once you do that, you are going to come over to the middle tables right here. Okay. And there's going to be this ice bucket right here. And inside the ice bucket, there's going to be ice and also ethanol. Okay. Ice cold ethanol. So what you're going to do is you're going to grab these transfer pipettes. Okay. These are for the ethanol only, so please don't throw this away, and don't use these for anything except for the ethanol. So you are going to get 5 milliliters of ethanol, ice cold ethanol, using this pipette, the same technique. Draw up to 1 millimeter mark. milliliter mark. And you're going to put the ethanol, ice cold ethanol, into the test tube at a 45-degree angle. So you hold the test tube at a 45-degree angle, and you put in slowly 1 milliliter, 2 milliliter, 3 milliliter, 4 milliliter, 5 milliliter. Okay. Then put the pipette back, put the ethanol back, and you do not want to shake the solution in this test tube. What you want to do is you want to just kind of let the DNA solution just kind of condense out. Okay, and then what will happen is you will see that this kind of whitish string-like substance will start to float to the top. Okay, that is the actual DNA of the strawberry condensing. So what you can do to take it out is use this glass stir rod. Okay, and you can kind of like rotate like this and just kind of like scoop it out slowly. Once you do that, what you want to do is you want to take down your observations, like what the DNA looks like, touch it, how it feels, and just like the general appearance. Once you do that, you are done with the strawberry extraction part of this lab. Okay, so to clean up, you can just wash the DNA down the sink right there, rinse off the glassware, and that's it. Part 2 of the DNA extraction lab. So part 1 was the DNA of a strawberry. Now what we're going to see is DNA from our own cheek cells. Okay, so what you want to do is find, it says right here, salt solution for the cheek. Okay, so you want to make sure that you're not chewing any gum. If you had like stuff in your mouth, maybe like you still have pieces of like your breakfast, you might want to go to the bathroom and like just kind of like wash out your mouth. Okay, come back. You're going to open this. You are going to put the contents, the salt solution, in your mouth, but not swallow. So don't swallow this because this is kind of a pretty salty solution. And so you're gonna put it in your mouth, and then you're going to kind of gargle, mix it around, like that. Use your teeth to kind of scrape the side of your inside of your cheeks while you're mixing the liquid inside your mouth without swallowing. Okay, you don't want to draw blood. We don't need the blood. We just need the cheek cell. So you're going to be doing this for about a minute, and then once you're done, you're going to spit it back everything into the same cup. Okay, and then once you do that, you are going to get a transfer pipette, a clean transfer pipette, and you are going to grab one of these conical tubes that says detergent solution. Okay, so this detergent solution, you are going to add 3 milliliters of the salt solution from your cheek, and also that has your cheek cells in it, into this tube that has already 5 milliliters of detergent solution. Okay, so you just open it up. Put in 1, 2, 3 milliliters. Okay, then you want to cap it. Okay, this transfer pipette you just throw in the trash, and then you want to kind of like mix it around for about a minute or so. Okay, the next thing that you want to do is you are going to use a centrifuge. Okay, so these are the centrifuges that we're going to be using in class. Okay, we actually have 3 of them set up, but I'm just going to show this one so it's a little easier. So you're going to open it up and make sure the conical tube is capped. Okay, and the thing about the centrifuge is you have to balance them. So if you look from the top, there are numbers 1, 2, 3, 4, 5, 6. Okay. If you only have 2 conical tubes, then what you want to do is you want to place them across from each other like that. You want to make sure they also have the same amount of liquid, or else what's going to happen is this is going to spin, it's going to be off balance, and it's going to start making a grinding sound. Okay, and that might damage the centrifuge. So make sure that these tubes are all the same volume. Okay, so put them across from each other like that. If you have 4, then what you could do is you could place it like that in that arrangement. Since we do have a pretty large class, we'll probably have to use all 6 slots. So like that. Okay. Once that's there, then you can close it up. Okay. And you want to set this to spin for about 3 minutes. So there's like a 3-minute number right there on the dial. Close it like that. It'll spin up. And then we just let it spin for 3 minutes, and it will automatically stop on its own. So like let's say it hits that time. So don't lift it up even if you hear the thing. You want to wait for the centrifuge to stop spinning the tubes. Okay, so it just takes a while to stop spinning. And then the reason why we centrifuge the DNA is so that we can separate the DNA from all the other stuff inside the cell. So the centrifuge spins it at a really fast rate so that like the heavy things inside the DNA solution will sink to the bottom, and the more lighter things such as DNA and proteins will float to the top. Okay, so once this thing has completely stopped moving, then what we're going to do is we're going to open this up, and then we're going to take our tube to where the ice-cold ethanol is in the center of the classroom. Okay, so as you can see, it just takes kind of a while for the thing to stop spinning. Please be nice to your classmates and actually wait for them to bring the tube so that we can actually fill this thing with 6 test tubes or conical tubes before spinning it. Okay, so grab yours, and then come over to where the ethanol is, and you're gonna grab ice cold ethanol from the ice bucket. Okay, and you're gonna get 3 milliliters. 45-degree angle. So again, the conical tube that you're putting the ethanol in should be at a 45-degree angle. And you're going to put 3 milliliters of ethanol in this. So 1, 2, 3, like that. You want to put the ethanol back in the ice bucket. And just like with the strawberry DNA, you're going to notice the DNA in the conical tube is going to float to the top. Okay, so once it does that, you're going to use a glass stir rod, and then spool out the DNA. And just like with the strawberry DNA, you want to write down your observations, touch it, feel it, compare it to the strawberry DNA, both the color, texture, feel, and also the amount. Once you're done with that, you can throw away this conical tube, rinse off the glassware, put away your equipment, and you're good. Thank you.